So here at IMW 2019 in Boston, one of our abstracts using so-called immunopaths was selected for an oral presentation, and I gave that talk. This is work I've done together with my colleague Gary Ulaner and colleagues at Sloan Kettering in New York. We had based all this work on work we started off in the lab. We took a monoclonal antibody targeting CD38. In this case we used daratumumab because it's FDA approved. We then ligated that with a DFO chelator, and then we labeled that with zirconium-89. That allowed us to inject this into mice and have CAT scanners from mice in the lab, and we could actually see myeloma because the antibody binds to the myeloma. So we had mice carrying human myeloma, and then we injected antibody, and we could then see that in the mice. That led us then to develop a clinical protocol, a phase 1 study. So this is the first ever, first-in-man phase 1 study in multiple myeloma with immunopaths. So I showed the results here at the meeting from this study, and we show that it's a safe approach. You can also see myeloma in patients. You can see where the myeloma is located. It corresponds with CT destructions. And this phase 1 study, because it's phase 1, was focusing on safety and feasibility, did not have a lot of results for the purpose of sensitivity and detection compared to other modalities. That will be done in a phase 2 study. But there was one patient that we had imaged with conventional FDG PET-CT within a week after this immunopath. And the FDG PET-CT was completely negative. And then when we did the immunopath in the same patient a week later, we show that there are multiple sites of active uptake. When we looked at the CT, we saw that these sites also correspond with lytic destructions. So we did not have, at this time, information from image-guided biopsies from the site, so we don't know if it's myeloma, although the CT strongly suggests that it is. We're looking into that. But overall, this very supportive, these supportive results from the phase 1 trial has prompted us to go forward. So we are now planning on opening a phase 2 study that will compare conventional FDG PET-CT with immunopaths, and also to compare blood tests with M spikes and light chains, as well as bone marrow biopsies to this test. And the intent is also to do image-guided biopsies of these sites to try to take out the cells and to sequence them. So this could be a new way of detecting potential residual disease that you would not see with blood tests, you would not see with imaging, and you would miss if you did a conventional biopsy, because it could be a matter of just having 1 or 2 residual sites that may not even be in the bone marrow. So this is very exciting.